Yeah, thank you very much. Thank you for having me. And yeah, I can just continue actually where Leo Trasande stopped, because he's told us that $249 billion dollars per year are caused by chemicals in plastics. But these are only a few of the chemicals we have in plastics. 16,000 were identified in the recently launched Plaschem report, and they comprise a wide range of intentionally added substances with different functions, but also the non-intentionally added substances. And these are formed during the production or are impurities of starting substances. And in this Plaschem report, around 60 were identified. But actually, the bulk of chemicals in plastic products likely consists of non-intentionally added substances, but we just don't know them. And here we have clearly a knowledge gap. But also of the 16,000 known plastic chemicals, more than 10,000 lack basic data on identity and toxicity, while more than 4,000 are considered hazardous. So that means that the composition of finished plastic products is just largely unknown, and consequently its toxicity too. As these compounds are mostly not covalently bound to the plastics, they can migrate, and humans can be exposed to them, for example, via the consumption of food that was packaged in plastics. And as we don't know the chemical composition in the plastics, we also don't know what actually is migrating and into the foods. But of the few toxic plastic chemicals we know, what we just heard this morning over, is that many are endocrine disrupting compounds and also metabolic disrupting compounds, which have been gaining more attention recently, and we heard about this morning. And the nuclear receptors I focus on in the endocrine and in the metabolic system are the peroxisome proliferator receptor gamma, which is important in adipogenesis and linked to obesity and associated metabolic diseases. And also then the pregnant X receptor, which is mostly known as xenobiotic sensor, but is also increasingly recognized as being involved in regulating energy homeostasis, for example, lipid and glucose metabolism. So these are some pathways that we know about that have been looked into more closely, but this, as also I think, this morning in the metabolic disrupting talk was said, we have so many different pathways and potential receptors and so many different chemicals. So it is likely that we have other pathways also activated. And one large group of cell surface receptors are the G protein coupled receptors. They transduce more than 80% of cell signaling in the body and have important physiological functions. 35% of the FDA-approved drugs actually target GPCRs. But there are just almost very few studies looking into the industrial chemicals interfering with these receptors. One example, a prominent example that there is the G protein coupled estrogen receptor 1, which is BPA activates this one. So yeah, we have talking about this whole morning. BPA is an important chemical, but also a few others where we know that they can interfere with a range of cellular receptors. And this has led to some restrictions in food contact materials, BPA, for example. But yeah, we have many more chemicals in plastics. And yeah, here we just don't know what they actually do. And so the overarching aim question I'm addressing here is, if we are exposed to additional endocrine and metabolic disrupting compounds coming from everyday food packaging. And one way to address these mixtures of unknown chemicals is by using effect-based tools, because they integrate the toxicity of all the unknowns and also possible mixture effects. And then we can combine this with an on-target chemical analysis to also look at the chemical landscape of these plastic products. And what I'm going to show you here is 4 studies. One part looking at nuclear receptors, the one that I showed you before. And first to look at the worst case scenario, so which are the chemicals in these plastic products and what is their endocrine and metabolic activity. To then expand our notion of different receptors, looking into the GPCRs as additional targets of plastic chemicals. And follow then another line and see if they actually also migrate under more realistic conditions into food packaging. And the last one is, we attempt to identify the toxicity-causing compounds in these mixtures. So, to this aim, we chose 36 food contact materials from bottles, cups, made of the 7 polymers with the highest market waste share. And we got them from 5 countries with a high plastic waste generation per capita, so that we have just a global picture of what consumers actually use. In the first studies, we used these methanolic extracts. We had them for 1 hour in an ultrasonification. Then this went directly to a non-target high-resolution mass spectrometry for chemical fingerprinting and tentative identification. And the others were analyzed in the Calux reporter gene assays with these 4 nuclear receptors, so the 2 peaks R and pipa gamma of the metabolic system, and the 2 sex steroid receptors, estrogen receptor alpha and androgen receptor, that was analyzed in antagonistic mode. So, jumping directly to the results, I told you that we have many chemicals used in the production of plastics, and we found 60,000 unique chemical features, whereby a chemical feature is accurate molecular mass associated with a chemical compound. We have here ordered according to the polymer type, the samples, and these bars indicate the number of chemical features. So we have a large diversity between 40 and almost 10,000 chemical features in just one sample. Looking now at receptor activation, here peaks are color-coded, where we have EC20 values. Blue is no activity, and the darker red, the more activity. We see that all but 3 samples contained chemicals that activated this receptor, and this reflects its role as xenobiotic sensor that can bind structurally very diverse chemicals. Pipa gamma was activated by more than half of the samples, and the anti-androgenic activity and estrogenic activity we found in around 1/3 of the samples. And we also had some that induced cytotoxicity, which were then diluted to test for the receptors. What you can see here is that the samples that were that did not activate the peaks R, they also did not activate any of the other chemicals, and all had lower numbers of chemical features. So it shows us that this is, actually we found a correlation there between the number of features and peaks R activation and the other receptors except estrogenic activity. Not shown here, but we did not find any differences between the countries. So independent in which country, from 3 different continents with different legislations, where you can always buy food packaging with endocrine and metabolic disrupting compounds inside. But where we found a difference is between the polymer types. So polyurethane, PVC, contained more chemicals or activated more receptors. And also LDPE, low-density polyethylene, which was more surprisingly to see this strong. I'm looking a bit closer now into the differences between the polymers. Here we have pipa gamma, EC20 values, and the different polymer types. So I put the samples of one polymer type together. And we found that low-density polyethylene and PVC induced significantly stronger pipa gamma activity. I mean, the chemicals in there, as compared to PET and polypropylene samples. Estrogenic activity was significantly stronger induced by chemicals of the polystyrene, with 4 out of 5 samples activating this receptor very strongly. And we found the same for pellets, indicating that have less additives, for example, so indicating that this has really probably something to do with the polymer. Cytotoxicity was significantly stronger in low-density polyethylene samples and PVC samples. Both of these polymers are known to be more toxic. They are toxic starting substances, monomers, and also polystyrene is known to be more toxic. So we confirmed this. LDPE, yeah, we were not aware of this before. So yeah, so we found in these extracts, we found a lot of chemicals and we found a lot of biological activity. So yeah, so it is quite likely that we have other targets of these plastic chemicals. And that's where we looked into the G protein coupled receptors. This is a study led by Molly McPartland, and we looked into 126 GPCRs that are important in transmembrane signaling, important drug targets, but we know very little of them. We used the same extracts this time, as we had so many receptors, we needed to reduce the number of samples. So we compiled a PT mix, a PVC mix, and an assorted mix with the other polymer types in there. And we also tested some single chemicals that have a high prevalence in human samples. So in the first screening, I don't want to go into detail here, but these are all these interactions. And we have a cluster analysis here where red indicates receptor activation. So we found that 10 of these GPCRs were activated by the chemicals we tested, or the mixtures we tested. And the most pronounced activation was found of the adenosine 1 receptor and the melatonin receptor. Both are known to be to regulate the circadian rhythm. An example of an adenosine antagonist is actually caffeine. And melatonin is also known to be involved in regulation of insulin secretion. So we had these several mixes that activated them, and we wanted to know which are the products that the plastic products that actually activate them. So we tested them separately and found that, polyurethane and PVC were activating, and here also PVC in different potencies were activating these receptors. So this confirms our notion that we have PVC and polyurethane are more problematic. As GPCR signaling is very complex, because we can't directly deduce any health implications besides that regulation of circadian rhythm and insulin are associated with non-communicable diseases, but you can't just draw this conclusion. So here we need further downstream research that we just talked about this. But so concluding this first experiments with the extracts, we saw that there was large chemical complexity, that most of the plastic food packaging articles contained endocrine and metabolic disrupting compounds, and also several activate GPCRs. But these were extracts. So, do they actually, does this matter? Do they migrate into foods we eat? And that's what we wanted to check in the next study. So we used food simulants according to EU regulation, as a best case water, and then a simulant for dairy products like yogurt or milk, water ethanol mixture 1:1. And so we prepared out of 14 food contact articles. They were migrated for 10 days at 40 degrees according to regulation, and then concentrated with the solid phase extraction and analyzed in the non-target chemical analysis, and with the same 4 nuclear receptors. Jumping to the results here. So just let me check how my time is. Okay, so here I won't go into detail. but the question was like, do they migrate? And yes, they do. We can see clearly that these metabolic and endocrine disrupting compounds migrate into water. We have here color-coded again, with more red, the more activity. And we see the same pattern. So PXR was again activated by most of these migrates, and also the same polymers were activated, pretty much as in the extract study. So looking at the chemical composition here, we found pretty similar numbers of chemicals between the methanol extracts and the ethanol water migrates. That was kind of surprising. Also, they had very similar abundances, but much less in water as we had expected, because most of the chemicals are not that good soluble in water. In total, also here, we found 17,000 chemical features, of which we could tentatively identify only up to 10%. So most of them we don't know. And we don't know which are actually the toxicity driving compounds. And that's what we wanted to look into in the next study. So we actually used a statistical approach, but what I'm showing you here is an experimental approach where we used a effect-directed analysis to identify the toxicity-causing compounds. So we have our toxic sample that we fractionated with an HPLC into 60 fractions. These 60 fractions were analyzed again in the in vitro assays. To then, if we have a toxic fraction, we identify the chemical or tentatively and confirm the identity and toxicity again of this compound, this candidate. We did this with 4 samples with different rates of success. I'm going to show you the estrogenic activity of this PVC tube, which had a pronounced receptor activation in the ethanol water migrate. So we had these 60 fractions, which you can see here. And this is the original sample. And we have one clearly estrogenic fraction here. And we could confirm this in the dose response. We were able to recover 51% of the activity of the original sample. So which one is the, what are the chemicals in this fraction? We did a non-target liquid chromatography and gas chromatography. And in the GC, we got one nice large peak, with a nice spectra, and which was tentatively identified as octylparaben, which is known to be estrogenic and also has been found in plastics. So we bought the standard, but could not confirm it. The retention time didn't match, but it looked very similar, and it was likely an isomer, found the molecular ion. And so the next best hit was ethylhexylparaben, also known to be estrogenic. So we bought this one, and this one time we were lucky and have identified it. We can confirm that it's ethylhexylparaben, retention time and spectra match. So next question, is it actually estrogenic in our assays? And we tested this in the dose response and found it to be estrogenic. What is now still one step we need to do is to quantify the concentration of ethylhexylparaben in the original sample and in the fraction to be able to explain how much does actually this compound explain of the toxicity. So taking this together, we used effect-based tools to look at the plastic food contact articles and identify endocrine and metabolic disrupting compounds, and could identify GPCRs as additional targets, and also showed that they actually migrate into food simulants, indicating that they are readily available for human exposure. We found that samples from across the globe were activated, and taking this together with all what we learned this morning, the widespread use of plastics, the adverse health effects we know from several plastic chemicals, we can't say that plastics are safe because they have so many unknowns, and we have all these receptor activation in there. Effect-directed analysis is a way how to identify toxicity-driving compounds, but it comes with challenges. And I showed you the best example. And if you're willing to invest 1 year of your life, that works, but we won't be able to do this for all the different products with different chemical compositions. One thing that would ease this a lot to identify the toxicity-driving compounds is using, if we would have more information on use, identity, and toxicity of the plastic chemicals, including in the non-intentionally added substances. And this is also a prerequisite for their current safety assessment, right? That we know what is in there. We found that the simpler chemicals are, the less receptor activity was induced. So this indicates like a redesign. We just need to make them chemically simpler. And we have some examples there. I didn't show you this, but there are some products, same function with less, and some with more chemicals and toxicity. Also, the toxicity-driving compound, this paraben, is known to be problematic. And in this Plasticchem report, parabens were identified as a priority group for redesign and for regulation. So this is also something that indicates that group-wise management could help, and we definitely need international efforts because we have these globalized production and supply chain. Thank you for your attention. Also, thanks for this great team from Martin Wagner, where this research was conducted, and to the European Union for funding. Thank you.